Throughout this lab section and what we have learned so far, a topic that was hard for me to understand or grasp was metabarcoding and all the steps and details it entailed because there's so much context to it. So what I did was obviously look over all the information I had from the course and also try to find different ways to understand metabarcoding. So I wrote down a few main ideas of what metabarcoding is. So that's what I'm going to explain. So metabarcoding is a technique that allows scientists to identify multiple species with an environmental sample by analyzing DNA sequences. Basically, it's like a genetic fingerprinting method for ecosystems, if that makes sense. So the process begins with collecting environmental samples such as soil, water, or even air. These samples contain traces of DNA from various organisms known as environmental DNA or eDNA. In this lab, DNA is extracted from all the samples. Scientists then amplify specific regions of a DNA using a technique called PCR, which focuses on the barcode regions, short standardized segments of DNA that vary between these species. Next, the amplified DNA is sequenced, producing millions and millions of DNA sequences. These sequences are then compared to reference databases to identify the species that is present in the sample. This metabarcoding has a wide range of applications. It helps monitor biodiversity, assess ecosystem health, detect invasive species, and it also studies the diet in animals. It also offers very advantages. It's faster and more cost effective than the traditional methods. However, it also has its limitations, which it can sometimes miss rare species or produce ambiguous results if the reference databases are incomplete. So in summary, metabarcoding is a tool and ecological research offering insights into the complex web of life on Earth. And that's it. Thank you for watching.